All right, guys, this is Unit 1, Chemistry of Life, Chapters 2 and 3, review for summer assignment in preparation for the test. So, the topics, first I'm going to cover our structure of water and hydrogen bonding, the elements of life, biological molecules, those major organic compounds, carbs, lipids, proteins, and nucleic acids, both their structures and functions, as well as talk about the functional groups, and then focus a little bit more on the specifics of nucleic acids. At the end of this, you should know the elements that are necessary for carbon-based systems to function, how and what conformations molecules called monomers bond together to form polymers, the property of water and how water plays a vital role in the survival of individuals and biological systems, the structure of monomers and polymers, and how that directly affects their function. One of the key themes of AP biology is to know that structure dictates function. Always remember that the structure is going to be directly related to the function of a molecule. And also, we'll talk about how living systems exist in highly complex organization that requires an input of energy and the exchange of those major organic compounds, so the carbs, lipids, proteins, and nucleic acids. So, first things first, what is the role of energy in the making and breaking of polymers, and how do living systems function with the polarity of the water molecule? So if you think about the making and breaking of polymers, there's two processes that you have to be familiar with, and that's dehydration, synthesis, and hydrolysis. And then also we need to know how water is related to both the dehydration, synthesis, and hydrolysis. Because if you think about those two words, when you think of dehydration, you should think of the removal of water. And when you think of hydrolysis, that's going to be the addition of water. So water is going to play a major role in how living systems are going to function, because whenever you are combining monomers to make polymers, or if you are breaking polymers down to the monomers that our body systems need to use, it's going to require either the addition of or the removal of water. So, I'm thinking of the chemical context of life. Organisms are composed of matter, and we know that matter is anything that has both volume and mass, and is made up of elements. An element is simply a substance that cannot be chemically broken down to another substance. So think of hydrogen, oxygen, helium. Those are all elements. You can't break them down to another substance that's going to maintain those properties. Now, they can be broken down to their subatomic particles, such as electrons, protons, and neutrons. But in breaking them down to their subatomic particles, you are not going to maintain the properties of that substance. So therefore, an element can't be broken down by any other means other than chemically, and it is going to had a little interruption there, but that's all right. So when an element is in its elemental form, something is in its elemental form, you can't break it down to another substance that is going to maintain those properties. Now, when we think of compounds, a compound is going to be something that consists of two or more elements in a fixed ratio. So water would be considered a compound because that is H2O, meaning there's two molecules of hydrogen and one molecule of oxygen. But if I had something such as hydrogen gas, which is H2, that is just elemental hydrogen because it's only made up of one type of element. So keep in mind, a compound is going to be two or more different types of elements, and they have to be in a fixed ratio. So O3 is elemental, but H2O is compound. Now, if we're thinking about sodium and chlorine, so sodium is an explosive solid by itself, and chlorine is a poisonous green gas by itself. So those are the elemental states of those two substances. But if you were to combine them together in a compound, you get sodium chloride, which is table salt. So typically what's going to happen with a compound is the properties are going to change once they come into a fixed ratio with another element. So sodium has properties, chlorine has properties, but sodium chloride has a completely different set of properties. So when you make a chemical bond, chemical bonds are going to change the properties of elements to compounds. Make sure that we know that there are 92 naturally occurring elements. 20 to 25% of those naturally occurring elements are what we call essential, and Organisms are going to be made up mostly of carbon, hydrogen, oxygen, and nitrogen. Those are the most important ones. Also phosphorus, because you have phosphorus in both DNA and your proteins. Trace elements are elements that are going to be required in minute amounts. So if you think of like iron, you need iron, you need manganese, you need chromium and cobalt, but you only need those in very small amounts. So those are considered trace elements. So we should all know the basic structure of an element and how to identify the mass number, atomic number, and things like that. But let's just go over it real quick. We know that the subatomic particles of an atom are protons, neutrons, and electrons. Protons have a positive charge. Electrons have a negative charge. Neutrons are going to have no charge. The job of a neutron is to basically act as a buffer in that nucleus with those protons, because we do know that like charges repel. So the more protons an atom has, the more neutrons they will need to serve as a buffer zone so that the protons don't repel each other and actually destroy the nucleus of the atom. Electrons are going to be found outside of the nucleus, unlike protons and neutrons, and electrons are responsible for the behavior of the atom. So electrons are going to determine the chemical behavior, and protons are actually going to determine the identity. No two elements have the same number of protons in their nucleus. So you can identify the identity of an element by the number of protons. If there's 1 proton, it's hydrogen. If there's 2, there's helium. Okay, if there's 3, it's lithium. So you can't have a lithium with 2 protons. Anything with 2 protons would be considered helium. If you're looking at the actual setup of the symbol for an element, you're going to have the symbol, which is Na for sodium. You're going to have the mass number, and the mass number is equal to the number of protons and neutrons, or the number of nucleons, which are the things found in the nucleus. The mass of an atom is determined by protons and neutrons. They each carry a mass of 1 AMU, or atomic mass units. The mass of an electron is so small that we say that it's nil, and it does not affect the actual overall mass of the element. Here at the bottom, you're going to see a smaller number. This is the atomic number, or the number of protons in an atom. You can also determine the number of electrons in a neutral atom, because the number of protons or positive charges will be equal to the number of electrons, so those negative charges. Isotopes are going to be 2 atomic forms of an element that have a different number of neutrons. Carbon comes in carbon's 12, carbon 13, carbon 14. And so carbon only has a set number of protons. Every carbon atom has 6 protons. However, if that carbon atom has 6 neutrons, 6 plus 6 is 12, so it's carbon 12. If it has 7 neutrons, it's carbon 13. Or if it has 8 neutrons, it's carbon 14. And we call those isotopes. So isotopes are going to be an element that has a different number of neutrons. Because the proton number remains the same, it is still determined, still to be the same type of element. If I have 7 protons, that is no longer carbon, that's nitrogen. But if there's 7 neutrons and 6 protons, I still have carbon, just a different isotope of carbon. Isotopes are important for dating fossils, for scientific experiments to trace metabolic processes, and also in diagnosing medical disorders. Typically, radioactive isotopes are used because of how they decay and how they give off different particles and energy, and they can be tracked to determine several different things in the medical field, and also the age of a fossil. Now, atoms form bonds to become more stable. The magic number is 8. For most atoms, they want 8 valence electrons, which are the electrons in the last shell. When atoms don't have a stable valence shell or last shell, they transfer or share electrons in order to satisfy that need for a stable valence shell. Based on how atoms become stable, we have different types of bond. The strongest type of bond will be a covalent bond, where electrons are shared. Ionic bonds would then be the next type of bond where there's actually a transfer of electrons from one element to another one. And when that happens, it forms an ion, because an ion is an atom that carries a charge, meaning there is an imbalance between the number of protons and electrons. So an atom that is going to give away an electron will have a positive charge or become a cation. And the atom that accepts the electron will carry a negative charge or be an anion. And because opposites attract, the cation is now attracted to the anion, forming an ionic bond, which is actually an attraction, but a very strong attraction. The next type of bond or attraction will be considered a hydrogen bond. And a hydrogen bond is going to be between a positively charged hydrogen in one molecule and a negatively charged oxygen or nitrogen in a different molecule. And this bond that's formed between that hydrogen and that oxygen or nitrogen is called a hydrogen bond, which is again more of an attraction between opposite charges, but it's very important for the properties of things such as water and DNA. Our last type of attraction, which is the weakest, is called van der Waals. And van der Waals, if you think of, they're more like hot spots because electrons are not static. They do move around. And as they're moving around their electron cloud or in those shells, they create momentary pockets of charge because there's different electronegativity in certain areas. And those pockets of charge can interact with pockets of charge in other molecules or within that same molecule, if it's something like, say, for instance, a protein, and they create temporary attractions which can help to stabilize the structure of a compound, a molecule, or a substance. And remember that structure dictates function. So we find van der Waals interactions are extremely important in things like the tertiary and quaternary structures of proteins because they help to maintain that structure. They're also broken very easily. And so with proteins, we can see that with the change in pH or temperature, those structures can be released and cause a denaturing or a change in shape of the protein. And so remember, if we change the shape, we change the function. As I've said before several times, structure dictates function. If you remember nothing else from this, remember that structure will dictate function. So we know that chemical reactions are going to make or break bonds. You have starting molecules, which are called reactants, and your final molecules, which are your products. Most chemical reactions are reversible. And once a chemical equilibrium is reached, that's going to be when the forward and reverse reactions are at an equal rate. That doesn't mean that the reaction stops. That just means that the reverse that is going forward and in reverse at the same level. So we've already talked about hydrogen bonding, how it is the bond between a hydrogen that carries a positive charge in one molecule and an oxygen or a nitrogen in a different molecule that is going to carry a partially negative charge. This is extremely important in the properties of water. Water is considered a polar molecule because the difference in electronegativity is so great that oxygen has a greater pull on hydrogen's electrons than hydrogen does. So the two hydrogens in a water molecule appear to carry a slightly positive charge, whereas the oxygen is going to carry a slightly negative charge. Now, these slightly positive charges in these hydrogens are going to be attracted to either a slightly negative nitrogen in a different molecule, or if you look down here, the slightly negative oxygen in another water molecule. This polarity that gives rise to hydrogen bonds is going to make water a very unique molecule. Each water molecule can make up to 4 hydrogen bonds with another water molecule. Now, because of this polarity that leads to the hydrogen bonds, water has 4 emergent properties. One is being that it is cohesive, meaning that it is a sticky molecule. Water can stick to other things. One of the things that water will stick to is another water molecule. But water will also stick to other substances. So when water sticks to water, we call that cohesion. But when it sticks to something else, we call that adhesion. Now, the combination of cohesion and adhesion working together creates what's known as capillary action, Or if you've ever taken a straw and like covered the end and then stick it in liquid, you'll notice that the liquid is able to climb up into the straw without you actually having to apply suction. That is caused by capillary action. Capillary action is the ability of water to climb up a thin tube, and that happens because of cohesion and adhesion. The adhesion is between the water and the tube, which pulls the water up the sides, and then the cohesion is between the water molecules that's between the sides because they pull each other up in that tube. Another product of cohesive behavior of water is surface tension. Water tends to form bonds with each other, these hydrogen bonds with each other, which creates a tension on the top of the water, which allows things like water striders or spiders to actually be able to sit on top of the water. Now, the next emerging property is going to be moderation of temperature. Temperature is going to be moderated in areas around water because water has a high specific heat capacity, a high heat of vaporization, and it also has what's called evaporative cooling. What that means is that it takes more heat to change the temperature of water by 1 degree than it does the areas around it. So water can absorb a lot of heat. So if you're in an area around water, it is going to be able to moderate that temperature by absorbing a lot of heat before that water temperature is going to be raised. It also has a high heat of vaporization, which means it takes a lot of heat to make that water begin to break those hydrogen bonds and start to vaporize or switch from a liquid to a gas. And because of that, as it begins to vaporize, it is going to absorb more and more heat. And as it vaporizes, it is going to cool the area around it. So as water switches from liquid to gas and begins to evaporate, because it is absorbing the heat to break the hydrogen bonds, the areas around the water are cooler. So it's not sweating that actually cools you off in the summertime. It's the evaporation of the sweat. So, Mississippi problem. Mississippi is typically humid, which means your sweat does not evaporate. So, question, are you being cooled or are you just becoming a sticky, wet mess? Another emergent property of water is called expansion upon freezing. Ice floats because as water goes from a liquid to a solid, the hydrogen bonds become stabilized. In liquid water, the hydrogen bonds are constantly breaking and reforming, breaking and reforming. But as it goes from a liquid to a solid, they actually have to spread out and stabilize into a set position. Because it spreads out, it increases the volume of water. So as water freezes, the volume increases, but the mass stays the same. And we know that density is a property of mass over volume. So if the mass stays the same, but the volume increases, that is going to decrease the density. Anything that has a density less than that of liquid water will float. So because solid water has a smaller density, ice floats, which is a good thing because it is able to form a barrier in oceans and streams and rivers for aquatic life, which prevents the temperature of water from dropping too low in areas where it becomes cold in the wintertime. So it forms a barrier across the top of the ice because water freezes from the top down, which allows aquatic life to continue to live below the ice. The last emergent property of water is that it is a versatile solvent. And what we know from water, because it is polar, it carries slight charges, and like dissolves like. So because of its polarity, water is able to dissolve anything that carries a charge. So ionic compounds and compounds that are also polar, water will interact with. We say those things are hydrophilic because they allow water is able to interact with them and actually break those bonds. But anything that is nonpolar, water is not able to interact with, and we said those things are hydrophobic because water can only dissolve things that carry a charge or a slight charge, so ions and polar molecules, but it cannot dissolve nonpolar molecules. Remember, like dissolves like. So, most chemical reactions in organisms are going to involve solutes dissolved in water, and it depends on the molarity or the concentration of solutes in a volume of an aqueous solution. Again, hydrophilic substances have an affinity for water, whereas hydrophobic substances do not. So if we think about our sodium chloride, which is an ionic compound, meaning that there has been a transfer of electrons from sodium to chlorine. Sodium carries a positive charge, chlorine is going to carry a negative charge. So the water molecules are able to separate the sodium from the chlorine by interacting with the charges on the compound. So the water is able to form a bond around the sodium with the oxygens because oxygens are carrying a slightly negative and the sodium is carrying a positive charge. And the opposite is true for the chlorine. Since the chlorine carries a negative charge, it's the hydrogens on the water molecule that will interact with the chlorines. So solutes called acids and bases disrupt the balance between the hydroxyl and the hydroxide in pure water. This is how we end up with what's known as our pH scale. pH, or percent hydrogen, is going to be a measure of the number of hydrogen ions in a solution. Now, if there is an increase in the hydrogen ions, we say that is acidic, and that there is a decrease in the hydrogen ions, we say that that is a basic solution. A before B, acids before bases. Acids are going to donate a proton to solution or increase the hydrogen ion concentration, and bases are going to accept a proton from solution, which is going to decrease the hydrogen ion concentration. So what we need to know is that pH is a measure of the opposite log of the hydrogen ion concentrations. Acidic solutions have a pH value which is less than 7, and basic solutions are going to have a pH value which is more than 7. 7 is considered neutral. Now, a buffer is a substance that will actually minimize the changes in the hydroxide ion and the hydrogen ion concentration in solutions. So what a buffer does is it either accepts hydrogen ions or accepts protons or donates it. If the pH of the solution becomes too low, it will accept hydrogen ions to increase the pH. If the pH becomes too high, it will donate hydrogen ions to decrease the pH. Low pH, acid. High pH, basic. So how do we as humans affect the chemical context of life? One thing is that we burn fossil fuels. We take away trees, other things that can actually remove the carbon dioxide from the air. So just make sure you read over this slide. It talks about the ocean acidification and how that actually happens as there's an increase in hydrogen ions. It's actually going to combine with the carbonate ions, which forms bicarbonate ions. And the carbonate ion concentrations are going to decrease. And that decrease in carbonate ion concentrations is not a good thing, because then you're going to have a problem with the coral reefs, because the coral reef is made from the carbonate ion concentration. So if you don't have enough carbonate ions, then the coral reef is going to begin to die. And we do know the coral reef is like the rainforest of the ocean. It's where we have the most diversity in life for the aquatic animals. So that's it for chapter 3. And let's talk about, chapter 2. Now let's talk about chapter 3, which is carbon and the molecular diversity of life. So we do know that anything that is considered organic is going to contain carbon, and it usually contains hydrogen as well. Aside from water, living organisms consist mostly of carbon-based compounds. So your carbon-based compounds are going to be more your carbohydrates, your lipids, your proteins, and your nucleic acids. And again, remember that structure dictates function. So the structure of your organic molecules is going to determine how they are recognized, interact with, or respond to other molecules and each other. Now, carbon is considered the compound that is going to be important for organic substances because of carbon's versatility. Carbon has 6 electrons, 4 of which are valence electrons. Because of those 4 valence electrons, carbon can form 4 covalent bonds. Remember, the magic number is 8, so it's trying to get to 8 valence electrons. Carbon can form single bonds, double bonds, or triple bonds. And most often, carbon is going to bond with another carbon, hydrogen, oxygen, or nitrogen. In doing so, especially with the carbon-carbon bonds, carbon is able to form skeletons that are either chains, branch chains, or rings. Now, carbon can be arranged in these chains, straight chains, or rings, which means that carbon can create isomers. And unlike an isotope, an isomer is going to have the same number and types of atoms, but the structure is going to be different. And remember that structure dictates function. So if the structure is different, then the properties will be different as well. You have 3 different types of isomers. You have structural isomers, and then you have cis-trans isomers and enantiomers. So a structural isomer is simply when you can have like maybe a chain or a branch chain. So if you look at this example, you have 5 carbons in a row, and with those 5 carbons, you have 12 hydrogens. So the formula for this will be C5H12. Well, this is a straight chain. Now over here, I still have a C5H12, but in this case, I have a 2-methylbutane because instead of being 5 carbons in a straight row, so it's not a pentane, there's only 4. And coming from one of those carbons is a methyl group. See this functional group right here? This methyl group. And because of that, this is a different molecule, which will have a different set of properties. But because the formula is still C5H12, these are considered structural isomers, where the only difference is the arrangement of the atoms around the carbon-carbon bonds. Now, our next type of isomer is called a cis-trans isomer. So cis-trans isomers are going to be what happens around a double bond of a carbon-carbon atom. You can't really see it here, but this is a carbon with a double bond to another carbon. Now, these are considered constituents. So these are things that are actually bonded to the carbon that are coming off of this carbon-carbon bond. The location of the 2 constituents is going to determine whether or not this is cis or trans. Cis means same side. So if those 2 constituents are on the same side of the carbon-carbon double bond, because you cannot rotate a double bond, this is a fixed structure. This was a single bond, you can rotate it, but since it's a double bond, you cannot. So if they're on the same side of the double bond, we say that is a cis form. But if they're on opposite sides, then we say they are trans. So let's say that these 2 elements were lithium. Okay, this lithium would be on the same side. The formula would still be C2Li2H2 for both of these. However, the location of the lithiums will make this one cis and this one would be trans. We see the difference in cis and trans a lot of times in medications and stuff like that. And it is important because you can have an active form and an inactive form, just like when you have enantiomers. Enantiomers are mirror images of each other. It's like having a right hand and a left hand. You have your L isomer and your D isomer, where the L is like your left hand isomer and your D is your right hand isomer. If you speak Spanish, I think that the word for right is like derecho or something like that. It starts with a D. No, remember that. If I'm right, I think I'm right. If I'm not right, don't tell me. But anyway, this is important. Your enantiomers are important because one of these would be active and one of them can be inactive, or one could be helpful and the other one can be harmful. So we see this a lot in medications where they'll have a molecule where one is a placebo and the other one is the actual medicine. They typically will use an enantiomer of that molecule, of that medicine. So the L may be the active form and the D may be the inactive. And so to do trials to make sure that the medicine actually works, they'll give one group the active and another group the inactive. The formula for the compound would be the same, but the arrangement around that central carbon would be different. If you notice here, here's your central carbon. We have the nitrogen, you have the amine group on one side on the D and another side on the L. These are mirror images of each other, which means that one version would be active and the other one would be inactive. Now, your functional groups, which we kind of touched on in class, would determine how a molecule actually works or how it affects function of that actual molecule that it is attached to. Functional groups are the most important molecules to life because functional groups are going to determine, again, the function, which is why they're called functional groups. And it can replace one or more hydrogens, which is bonded to a carbon skeleton. A molecule can have multiple functional groups. There are 7 important functional groups in chemistry of life. So 7 important ones for us here in biology. Amino group, which is your NH2 or NH3 positive if it's in its ionic form. Your carbonyl group, which is your C double bond oxygen. Your carboxyl group, which is your COOH. Hydroxyl group, which is OH. And if you think about it, your carboxyl group is going to be a combination of carbonyl, which is C double bond O, and hydroxyl, which is OH. Because this one is COOH. Your methyl group, which is very important in DNA methylation, which controls genes being turned on and off, is CH3. Your phosphate group, which is PO43 negative, you should remember that from chemistry, one of your polyatomic ions. And your sulfhydryl group, which is like a hydroxyl group, except instead of oxygen, you have sulfur. So instead of OH, you have SH. So look at these individually. Here we have our amino group, which is an amine. We see these in amino acids. The amine part in its ionized form is picked up a proton and it becomes NH3+. Our carbonyl group, which is going to be a carbon with a double bond to an oxygen. Now off of this carbon on either side, you're going to have a different element combined to it, either another carbon on both sides, carbon-carbon, or carbon and a hydrogen. And the location of this carbonyl group is going to determine whether it is considered an aldehyde or a ketone. A ketone, if it's within the structure, so this carbon is going to have a carbon bonded to both sides of it. That's going to be considered a ketone. But if it's at the end of the structure, we consider it an aldehyde. Think of it this way. Ketone is in the middle of the alphabet. So, the letter K is in the middle of the alphabet. Ketone starts with K. So if my carbonyl group is in the middle of the structure of a carbon-carbon chain, it's a ketone. Aldehyde is at the beginning of the alphabet, the letter A. At the beginning of the alphabet, aldehyde starts with A. So if my carbonyl group is at the beginning of the structure, where it only has one carbon attached to one side and a hydrogen on the other, then that is an aldehyde. Aldehydes and ketones are extremely important when we talk about our monosaccharides, because the difference between glucose and fructose is actually the location of this carbonyl group. One's an aldehyde and one's a ketone. Our carboxyl group, again, is a combination of an carbonyl and a hydroxyl group. We see these also in our amino acids. This is a carboxyl N. It's sometimes called carboxylic acid. It's called an acid because remember, acids donate protons. So this hydrogen is going to leave behind its electron, making it a proton, and it's donated to solution. So in its ionized form, this is C double O negative. A hydroxyl group is hydrogen and oxygen. So an OH group. Anytime you have a bunch of hydroxyl groups added to a compound, it makes it an alcohol. And they tend to end with all. So if I was to take this hydrocarbon and replace this hydrogen with a hydroxyl group, what used to be ethane is now ethanol, an alcohol which is present in alcoholic beverages. Methyl group, CH3, a carbon with 3 hydrogens around it. We see methyl groups in a lot of compounds. Methylation is extremely important for identifying substances in a component of DNA. So it's been modified, which means the gene is either going to be turned on and off. We'll see a lot of methylation throughout the unit. And our last group is our phosphate group, which you should be familiar with. The phosphate groups are seen in ATP. It's one of the 3 in the triphosphate. they can form high energy bonds with each other. You also see phosphate groups in your DNA and RNA as part of the backbone, because the backbone of DNA and RNA are both sugar phosphate backbones. Here we have a glycerol phosphate where the hydrogen has been replaced with the phosphate group. Phosphate groups are extremely important in many chemical reactions. We're going to see phosphorylation throughout, which is going to help with creating, transferring energy and with changing reactants into substrates. And sulfhydryl groups, which is sulfur and hydrogen, is important in things like protein structure, because we see what's called disulfide bridges, where one sulfhydryl group will form a bond with another sulfhydryl group, which makes a very strong covalent bond, which helps to maintain the structure. And again, structure dictates function. Whenever we have a sulfhydryl group, we classify that compound as a thiol. So, a couple things to think about. Polymers are long molecules consisting of many smaller building blocks called monomers. Cells assemble polymers through the loss of a water molecule. This is your dehydration reaction. Cells disassemble polymers through the adding of a water molecule. This is called a hydrolysis. And these processes are facilitated by enzymes. Enzymes are catalytic proteins which speed up chemical reactions. So if we look at this diagram, we see a dehydration reaction. In a dehydration reaction, you're going to take a monomer, or it's another polymer, and you're going to add another one to it. And anytime you are going to be joining molecules together, there's going to be a removal of a water molecule. So the hydrogen from one molecule and the hydroxide from a different molecule will come together to form H2O, which is water, allowing this molecule to bond with this molecule. Hydrolysis is the reverse of dehydration. So if water went out to join these together, then the water has to go back in to break them apart, where the hydrogen will be joined to one molecule and the hydroxide will be rejoined to the subsequent molecule. So water goes in to break it apart, water comes out to put it together. Dehydration builds, hydrolysis breaks. There are 4 major organic compounds: carbs, lipids, proteins, and nucleic acids. Let's look at those. A carbohydrate is going to be a substance that contains carbon, hydrogen, and oxygen only. There's usually 1 carbon for every water molecule. That's a set 2-to-1 ratio of oxygen to hydrogen. In monosaccharides, we have a 1 to 2-to-1 ratio. That ratio changes once you make polysaccharides, because remember, water is being removed every time monomers join together. Our prime examples of monosaccharides are going to be glucose, fructose, galactose. Our disaccharides are things like lactose, which is milk sugar, sucrose, which is table sugar, which is simply 2 monomers joined together. Polysaccharides can either serve a purpose for storage of energy or structure. In plants, for storage, we have cellulose. And in plants for energy storage, we have starch. In animals, our storage molecule is going to be glycogen. We store about a day's worth of glycogen in our muscles and in our livers. And in animals and fungi, our storage molecule is going to be chitin. Chitin is what makes up the exoskeleton of things like arthropods or some insects. So like if you step on a bug and hear a crunch, you're breaking the chitin. Or whenever you eat crab or lobster and you have to break through that outer shell that we call it, that's actually made up of chitin, which is a structural polysaccharide. Main functions for polysaccharides are going to be fuel. So it serves as a type of short-term energy storage. Those molecules contain the energy in the bonds, not the actual elements, but the bonds between the elements. You're going to have energy stored there, and then those can be converted to where the energy can be passed on to ATP, which is the molecule that the cells use for energy storage. Now, remember earlier I said there's a difference between glucose and fructose, where one's an aldehyde and one's a ketone. Glucose is what we call an aldose because it is an aldehyde carbohydrate. Remember, most carbs end in ose, so we call glucose an aldose. And if you notice, where the carbonyl group is at the beginning of the compound. So it's at the beginning, like the letter A. So this is an aldose. Now, fructose, on the other hand, the carbonyl group is in the middle between two carbons. So like the letter K is in the middle of the alphabet. So we said this is a ketose. Now, the formulas, the chemical formulas for both glucose and fructose is C6H12O6. But our bodies can more readily process glucose than it can fructose. In fact, fructose can only be processed by the liver. And if you give it too much fructose, then that causes problems where it has to actually store that because it can't break it down. Glucose itself can combine in two different structures. It forms what's called alpha or beta bonds. In aqueous solution, glucose is going to form a ring. So again, if you're going to have your glucose where it is actually coming from a linear structure to a ring structure, then in doing that, you're going to remove a water molecule, which means there's no longer that 1:2:1 ratio. However, when it does happen, when glucose bonds with other glucose molecules, it can form either an alpha bond or a beta bond. And that is all determined by the location of the hydroxyl group on the number 1 carbon. If the hydroxyl groups are cis to each other, meaning on the same side of the molecule, you're going to form an alpha glucose bond. If the hydroxyl groups between the number 1 and the number 2 carbon are trans, then you're going to have what's known as a beta conformation of that bond. Why is that important? Well, as your monosaccharides bond together to form these glycosidic linkages, whether or not it forms an alpha or a beta bond is going to determine whether or not we as humans or other animals can actually digest those bonds. That's the difference between glycogen, starch, and cellulose. One reason that plants are considered dietary fiber is because of the presence of cellulose. Now, cellulose is a structure polysaccharide found in plants. Cellulose has 1,4 beta linkages, okay? So that because of those beta linkages, our bodies cannot break down cellulose. Our bodies are equipped to break down alpha linkages. And if you look here at starch, you see these are 1,4 alpha bonds. So each one of these bonds are going to be cis. And because of these 1,4 cis bonds, we have the enzymes that can break down starch. So we can digest the starch. We also have these 1,4 alpha bonds in glycogen, which is the animal storage molecule. But cellulose has 1,4 beta bonds where you go from cis to trans. So these are trans bonds of each other. And we lack the enzymes to break down trans bonds. So why do we want the cellulose? Because it serves as dietary fiber, which means as it passes through your intestinal tract, because it cannot be digested, it actually scrapes the lining of the intestines, which stimulates a release of mucus, which helps the fecal matter or unwanted particles to actually exit the body. Now, in addition to dietary fiber, you must also have enough water. You can't be dehydrated. Just a little side note, because you need water to make the mucus, okay? So, our next type of major organic compound is the lipid. Lipids are not true polymers because they do not have a true monomer. A monomer is a repeating subunit. Lipids are made up of different components depending on the type of lipid that you have. If you have a triglyceride, which we are commonly called fats and oils, you're going to have a glycerol, that alcohol backbone, and 3 fatty acid chains, also called triacylglycerols. So this is a triglyceride, tri for the 3 fatty acids, and the glyceride for the glycerol. These are important fats and oils, okay? This is our main source of long-term energy storage. Now, on the other hand, you can have one of these fatty acid chains replaced with a phosphate choline head. If that happens, you're only going to have your glycerol, 2 fatty acid chains, and then that phosphate, which is going to create a phospholipid. This is the major component of the cell membrane. And there's more type of lipids, but we're only going to focus on 3. The third type of lipid is going to be composed of hydrocarbon rings. Typically, you're going to have 4 fused rings with attached chemical groups. And whenever you see these 4 fused rings, this is indicative of a steroid, of a lipid steroid. And these are components of the cell membrane, such as cholesterol, which helps to maintain the fluidity of the cell. And also, the hormones can serve as signaling molecules that travel throughout the body, like your testosterone and your estrogen. The linkages between the backbone, the glycerol backbone, and the fatty acid chains is called an ester linkage. And that ester linkage is going to contain a carbonyl group. You see this carbon with the double bond to the oxygen here? And you see the removal of water here, where you're going to have this hydroxyl group removed from here. I'm sorry, the hydrogen removed from the glycerol, and the hydroxyl group which is going to be removed from the fatty acid. Fatty acids can be either saturated or unsaturated. A saturated fatty acid is going to have all single bonds between the carbons, okay? Whereas an unsaturated fatty acid will have at least one double bond between those carbon molecules. So that's the difference between having animal fat and a plant fat, where you're going to see with your saturation or unsaturation. If it's an animal fat, then you're going to have a substance that tends to be solid at room temperature. And if it is a plant fat, you have a substance that tends to be liquid at room temperature. And the difference between the two is actually the presence or lack of double bonds. Now, our next type of lipid is a phospholipid. And again, phospholipids are what we call amphipathic because they have that glycerol backbone with 2 fatty acid chains attached and a phosphate head. Now, this phosphate group is a hydrophilic side because remember, water is polar. Water likes things that carry a charge or a slight charge. Phosphate, PO43 negative, carries a negative charge, so it likes water. Whereas your fatty acid chains are going to be hydrocarbons. And those are non-polar covalent bonds. And whenever you have a lot of non-polar covalent bonds, that is a substance that is hydrophobic. It does not like water. So amphipathic means that this molecule has a water-loving side and a water-hating side. So it helps with the formation of the cell because it is able to form a bilayer, which creates a boundary between the inside and the outside of the cell. So we see here it's called a phospholipid bilayer, where the hydrophobic parts are tucked into the inside and the hydrophilic heads are going to come in contact with the external fluid of the cell and also the cytoplasm, which are both mostly water. Last type of lipids, again, are steroids. When you see these 4 fused rings, which can have other constituents which are coming off of it, but the key is to look for these 4 fused rings, which is the basic carbon skeleton, and this is going to create the backbone for our lipid steroids. And we do have protein steroids as well, and they have a different structure. So when you see these 4 fused rings, you need to think lipid steroid. Amino acids are monomers for proteins. There's several different types of proteins, enzymes, structural proteins, storage proteins, transport proteins. There's hormones, which are proteins, receptor proteins, motor proteins, defensive proteins, and each have their own function. But the key is that your proteins are going to be made up of a combination of 20 amino acids. An amino acid is going to be built around a central carbon. That central carbon has 4 different things around it. You're going to have a hydrogen. You're going to have a carboxyl group on one end, and you're going to have an amine group on the other end. And the thing that makes them different, this is the backbone of all amino acids. The only thing that makes amino acids different are what we call the R group. This is the variable region, where depending on what is bonded to this carbon at the R group position is going to determine the specific amino acid. Polymers are called polypeptides or proteins. Proteins are typically going to be a combination of multiple polypeptides. The bonds between each amino acid is called a peptide bond. This is the basic structure of an amino acid. Please make sure you know that. You're going to have your carboxyl group. That's a hydrogen up there. You can't see it. You have your central carbon with the hydrogen and an R group, and then you have your amine group, which is going to have your nitrogen and your hydrogens here. This is a neutral form. If this was in its ionic form, which would be an aqueous solution, you would have NH3 plus on this side and COO negative on this side. So here you can see where an amino acid is going to form a bond with another amino acid, where the water is removed because this is a dehydration synthesis, and then this carbon is going to form this nitrogen. If you notice, whenever you have an amino acid, you're going to have an amine group on one end and a carboxyl group on the other end. So you have what's called an N terminus and a C terminus. This creates directionality, which means this compound has to combine in a specific manner in order to form this protein. Now, you can also notice there's going to be a repeating nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, until you reach that C terminus N. So keep that in mind when you are trying to identify something by structure and you want to know if it's a protein or not. There's 4 levels of protein structure. You have your primary, your secondary, your tertiary, and your quaternary structures. If you look here, your primary structure is simply the sequence of amino acids in order. Those amino acids, because of their R groups, can interact with each other. And when they do that, they're going to form one of 2 structures in your secondary, either an alpha helix or beta pleated sheets. Pleated sheets is kind of like when you take a piece of paper and fold it back and forth, back and forth, back and forth. And your alpha helix is like a spiral. Now, by doing this, you're bringing R groups closer to each other as well. And those R groups can, the bonds in your secondary structure are going to be hydrogen bonds, okay? So here you're going to have covalent bonds, which are called peptide bonds in the primary structure. In the secondary structure, you're going to have hydrogen bonds. Now, once the secondary structure is formed, this molecule can fold on itself even more in its tertiary structure. Your tertiary structure is where you're going to see these disulfide bridges, which were formed by sulfur residues coming from most likely a sulfhydryl group. You can have ionic bonds because some of your R groups are going to be ionized to where they're either anions or cations. You can have hydrophobic interactions and van der Waals interactions, or you can have even more hydrogen bonds. So this tertiary structure is going to form what we call a subunit. Now, a quaternary structure is simply going to be multiple subunits. So this may be one subunit, and you may have a different subunit or multiples of the same subunit. But when those subunits interact with each other, you have what's known as a quaternary structure. Not all proteins are going to have a quaternary structure. Some of them are only going to have up to a tertiary structure because it takes multiple subunits to make a quaternary structure. It's also important to note that these quaternary subunits can be the result of one gene being transcribed and translated, or multiple genes being transcribed and translated. If the subunits are identical, it's one gene, but if they're different, like what we see in our hemoglobin, where we have 2 different types of subunits that come together, then that's going to be more than one gene. Again, there are 20 amino acids. Make sure you know the difference between nonpolar, polar, charged amino acids. So here is a picture of the polar. These are these are nonpolar. These are the polar that are hydrophilic. These are electrically charged. They are also hydrophilic. So if the electrically charged and the polar are hydrophilic, what does it say about the nonpolar? All right, they're hydrophobic. And what do we say about structure? Remember that structure is going to always dictate function. So with protein structure, especially with enzymes, it is important that proteins maintain their shape. When a protein is subjected to a different pH or temperature, the shape can be altered. That's called denaturation. When a protein is denatured, it is no longer going to function as it should because structure dictates function. However, some proteins, not all, are able to be refolded, and that's called renaturation. So if a protein is unfolded, it's denatured. And if it can be refolded, it undergoes renaturation. Not all proteins can be refolded once they are damaged. One of the main, one way you can see the effects of a change in protein structure is with the disorder sickle cell anemia. Sickle cell anemia is caused by a single miscoded amino acid sequence. One base was incorrect, which coded for one wrong amino acid. That amino acid caused the protein to fold incorrectly. So the beta subunit is folded incorrectly. When that beta subunit combines with the alpha subunit in a quaternary structure, you end up with a hemoglobin or a red blood cell that is shaped like a sickle instead of shaped as an oval as it should be. Now this causes problems because now it can't carry as much oxygen, and it also is prone to clotting, which is going to lead to all of the different symptoms that you see in a person with sickle cell anemia. One wrong nitrogenous base led to one wrong amino acid, which led to the improper folding of the polypeptide, which in turn made the entire protein shape be incorrect. And remember that structure dictates function. Proteins have a diverse and wide range of function, and it accounts for more than half of the dry mass in most cells. There are several different classes of proteins, and each class of proteins is directly related to its function. Enzymatic proteins are very common. All enzymes are proteins, but not all proteins are enzymes. Please make sure you remember that. And most enzymes end with ACE. You also have defensive proteins, which we see in our immune system. We have storage proteins, transport proteins, hormonal proteins, receptor proteins, motor proteins, other structural proteins. And our last major organic molecule is going to be our nucleic acids. Nucleic acids are going to be composed of nucleotides. Nucleotides have three structures. It's going to be a nitrogenous base or nitrogen-containing base, which can either be adenine, thymine, guanine, or cytosine, or uracil if it's RNA. Thymine is only found in DNA. So these are either going to be your purines or pyrimidines, which are nitrogenous bases. You're going to have a sugar, a 5-carbon sugar, or a pentose. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. And you're going to have one phosphate group. One thing that you will notice is that ATP carries the same basic structure as DNA and RNA. The exception is that there's going to be 2 additional phosphate groups. So sometimes ATP is classified as a nucleic acid because it does contain a nitrogenous base, which is adenine. It does have a sugar, which is ribose, a pentose, 5-carbon sugar, and it has phosphate groups. Instead of 1, it has 3. So ATP is sometimes classified as a nucleic acid. Please keep that in mind. Your 2 main types of nucleic acids, are DNA, double-stranded deoxyribose sugar, thymine as a base. Confined to the nucleus, stores hereditary information and dictates. It's going to control the cell's activity by dictating which genes are going to be transcribed. RNA is single-stranded usually. There's more than one type of RNA. You have small nuclear RNA, messenger RNA, transfer RNA, and ribosomal RNA. The sugar is going to be ribose. The nitrogenous bases will have the same cytosine, guanine, and adenine, but uracil only. It has various functions in gene expression, and carrying instructions from the DNA to the ribosome is also important in actually making the ribosome. That's what ribosomal RNA and proteins combine to make the ribosomal subunits. So RNA has several different purposes. Again, the monomer is called a nucleotide, which contains the phosphate, the sugar, and the base. If I was to remove the phosphate group and just have the sugar and the base, I would call it a nucleoside. Okay, nucleotide contains the phosphate group. Nucleoside is just the sugar and the base. The polymers are joined by what's called a phosphodiester linkage. Remember, in lipids, we have an ester linkage, but in DNA, it's a phosphodiester linkage because there's going to be a phosphate group participating in this combination of the backbone. The backbone contains a phosphodiester linkage and is a repeating sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. This is going to be true for both DNA and RNA. The polymers are called polynucleotides. The monomer is a nucleotide. The polymer is a polynucleotide. The two sugars are either going to be deoxyribose or ribose. That's determined by the functional group that is added to the number 2 carbon. If it contains a hydroxyl group, we call it ribose. If the oxygen is not there, deoxy, we call it deoxyribose. So this is just a ribose that lacks an oxygen at the number 2 carbon. Your 5 nitrogenous bases are going to be adenine, thymine, guanine, cytosine, and uracil. You have 3 pyrimidines and 2 purines. So the way I remember that is that you cut pyrimidines. So cut the pie. Think of py as being pie, cut the pie. So cytosine, thymine, and uracil are going to be your pyrimidines, and they also only have 1 ring. Okay. Your purines are going to be Ag. So think of Ag as angel. Angels are pure. So Ag are your purines, and they're going to contain 2 rings. So adenine and guanine are going to be your purines. 1 purine is going to form a bond with a pyrimidine. The bond between your nitrogenous bases are going to be hydrogen bonds. So adenine can bond with thymine and DNA, or uracil and RNA, and guanine bonds with cytosine. These are your main differences between DNA and RNA. Another thing to remember about your polynucleotides is that whenever you have DNA, you're going to have anti-parallel strands, where one is going to go from 5' to 3' and the other one goes from 3' to 5'. When we're talking about the primes, what we're talking about are the carbons in the compound. There's going to be 5 carbons in your sugar. So we count the carbons as 1, 2, 3, 4, 5. So when we're doing the backbone, this is your number 3 carbon, this is your number 5 carbon. So the way that it's going to form the bond, because it has directionality, is that the 5' carbon is going to be here, 3' carbon is here. So if I have a double strand, one strand will go 5 to 3, the other strand will go 3 to 5, the 3' carbon will be at the top. Let's see if I can find a DNA real quick. Nope. Okay, so this is a DNA strand. So this strand is going from 5' to 3', which means the other strand, or the leading strand, the top of it would be the 5' carbon. The bottom would be the 3' carbon. Now, DNA is going to direct the synthesis of proteins by DNA being transcribed to RNA and RNA being translated to proteins. That's called the central dogma theory of molecular biology, okay? The fact that DNA is transcribed to RNA and RNA is translated to proteins. So the gene that is transcribed, or the written message that is written as messenger RNA, is going to determine the protein that is going to be actually expressed or created, that's going to be created by the ribosome. We now know the sequence of the human genome that was completed in the early 2000s. And we are able to study things such as genomics, proteomics, and molecular biology simply by knowing that human genome. Genomics is used to analyze large sets of genes or compare genomes of different species, and proteomics is used to analyze sets of proteins. So think of that way. Genomics analyzes large sets of genes and compares them, and proteomics analyzes large sets of proteins and compares them. And that is it. You're probably going to have to replay and back it up and rewind it, because I'm pretty sure I was talking pretty fast, but hey, at least it wasn't in class. Toodles.